Hello friends and welcome to CEC live lectures. In the ongoing lecture series on extraction of natural products, till now we have discussed about the basic principle behind the extraction, what are the various factors that affect the extraction process, characters of a solvent, and what are the conditions that are required for appropriate extraction. Then we discuss about the conventional extraction techniques like maceration, decoction, infusion, digestion, refluxing, soxylation and percolation. We also discuss about their advantages and limitations. Looking into the limitations of the conventional technique, we discussed that various modern techniques have been developed, and then we discuss various such techniques like microwave assisted extraction, ultrasound assisted extraction, pressurized solvent or pressurized liquid extraction, supercritical fluid extraction, pulsed electric field extraction. In these techniques, we discuss about their brief historical development, the principle behind these modern techniques, how they are better than conventional techniques. We also highlight about the instrumentation aspect of these techniques, both at laboratory scale as well as at the industrial scale. Then we discuss about the factors that affect the extraction efficiencies of these techniques, followed by the application of these modern extraction techniques. We also discuss various methods that are or various techniques that are used to extract volatile oils from the plant matrix. So friends, in continuation with the lecture series, today I am going to start with another extraction technique named as enzyme assisted extraction. Now, as the name indicate, enzyme assisted, this extraction technique involves the utilization of appropriate enzyme to enhance the extraction efficiency of the process, and not only the extraction efficiency, but also the quality of the extract that is obtained after extraction. So, in this lecture we are going to discuss about the components of the plant cell wall, since these enzymes will be acting upon the cell walls of the plants, and as we discussed in my previous lectures that cell wall is the primary barrier that prevents the entrance of the solvent into the plant cell and thereby causing a limitation factor for the extraction process. So, in order to understand the utilization of different types of enzymes in this extraction technique, we first have to understand the components of the cell wall that protect the cellular components from extraction or prevents the cellular component from extraction. Then we will discuss what is exactly the enzyme assisted extraction technique. I will just give you a brief introduction about this technique, followed by the principle behind this technique. Then we will discuss what are the different types of enzymes that are used under this technique, followed by the steps or the process of this important extraction technique. After that, we will discuss various advantages as well as limitations of this technique, followed by what are the factors that can affect the efficiency of this enzyme assisted extraction process. Now, the beauty of this process is that it can be combined with various other extraction techniques, whether they are conventional or they are modern techniques. So, we can combine or we can extract a plant material by using a combination of enzyme assisted extraction vis-a-vis any conventional like maceration or percolation, along with any modern technique like microwave assisted, ultrasound or supercritical fluid extraction in combination with enzyme assisted extraction to enhance the overall extraction efficiency. And in the end, we will discuss about the application of this important extraction technique. So, first coming on to the components of the cell wall. I am sure we know that majority of the plant cell wall is composed of mainly polysaccharides, and according to an estimate, it has been found that more than 90% polysaccharides constitute the composition of the cell wall. Now, there are different types of polysaccharides that are involved in the structure of a cell wall, and these are namely cellulose, hemicellulose and pectins. Apart from polysaccharides, there are non-polysaccharide units such as lignins, as well as there are various other structural proteins that help in enhancing the ability of the cell wall to handle the mechanical stress or any kind of biotic or abiotic stress, or they also improve the mechanical strength of the cell wall to fight against various pathogens. Now, if we look into the structure of the cell wall, you will see that all these components are cross-linked with each other, and this is the beauty of the nature, wherein all the cross-linking of all these different components give additional or provide the required strength to the cell wall in order to protect the cell. Now, we will discuss each and every component of the cell wall. So, coming on to the first polysaccharide, that is cellulose. As we discussed that polysaccharide constitute more than 90% of the total cell wall composition, and amongst the polysaccharides, cellulose is the major polysaccharide that helps in the formation of the cell walls. And since it is a major component, it is considered as the hallmark of vascular terrestrial plants. Now, we know that cell wall is responsible for the protection of the cell or the cellular ingredients, and cellulose provides the mechanical strength to the cell wall of the plant cell. Now, if we look into the chemistry of cellulose, we will see that cellulose is a homopolysaccharide. Now, if a polysaccharide is made up of single monosaccharide unit, same monosaccharide unit, then we call it as homopolysaccharide, and if it is made up of different monosaccharide units, then we call it as a heteropolysaccharide. So, cellulose is a homopolysaccharide which is made up of multiple glucose subunits, and these glucose subunits can range from 25,000 to 1 lakh units that are attached with beta 1,4 bonds to give rise to a long glucose chains, which we also call them as glucan chains. And such kind of arrangement leads to high degree of polymerization of the cellulose molecules. Now, if you look into the structure of the cell wall, you will see there are different components. Now, these glucose chains or glucan chains, they are arranged in a specific manner which provides amphiphilic structure to the cellulosic chains that are present in the cell wall. And as we discussed that these are nothing but the glucose chains. So, and we know that glucose have hydroxyl groups on in its structure. So, these hydroxyl groups are present towards the outer side of the chain, thereby forming the polar surface or the outer surface as a polar surface of the cell wall, whereas the inner region is considered as the non-polar region of the cellulose or the glucan chains. Now, these glucan chains are interconnected to each other with the help of hydrogen bonds, and this interconnectivity through hydrogen bonds help in forming specialized structures of these glucan chains, which we call them as cellulosic microfibrils. So, we can say that cellulose is a homopolysaccharide which is made up of glucose units, and these glucose units are present in the form of long chains that are attached with the help of beta 1,4 bonds, and these chains are interconnected with hydrogen bond, giving rise to a microfibrillar structure towards the cellulosic arrangement. Now, if you look into the structure of this cell wall, you will see an orange thread-like structure that passes through these cellular microfibrils, and this orange thread-like structure is represented by B. So, this orange thread-like structure is the representation of hemicellulose, which forms the cross-linking between these cellulosic microfibrils. and this cross-linking gives additional strength to the cell wall. This also prevents the collapsing of these cellulosic microfibrils. Then in the figure you will see there are few proteinaceous structures that are labeled C, and these are nothing but the structural proteins that provide additional strength to the the plant cell wall. Then we have additional structure which is represented by D, and it is in a green color, again thread like structure, which are nothing but the pectin filaments that again cross link between the cellulose and hemicellulosic structures, providing additional strength to the cell wall. Then you see certain chemical structures that are represented by E in the figure. Now these are certain components that form bonds with these cellulosic and hemicellulosic or pectin structures, and thus such kind of chemical compounds or the secondary metabolites that form bonds with the components of the cell wall are very difficult to extract by using the conventional extraction techniques or even the modern extraction techniques. So once the extraction is complete, whether you are using a traditional technique or a modern technique, these compounds still remain intact with the cell wall components and are not extracted. Or in simple words, we can say that huge amount of such compounds are wasted in the biomass that is left after extraction. Now here comes the role of enzyme assisted extraction, which we are going to discuss ahead in detail. Now apart from these chemical structures that are represented by E in the figure, you can also see certain yellowish components that are labeled as F. These are nothing but the lignin structures that are interacting with the hemicellulosic or the cellulosic components, which provide additional mechanical strength to the cell wall and also help in preventing the water loss from the cell wall. So this is just the basic structure of a cell wall. Now we move on to the second major component, that is hemicellulose. So as we discussed, the cellulose is the major component or major polysaccharide present in the cell wall. It is followed by hemicelluloses. Now unlike cellulose, hemicelluloses are heteropolysaccharide. It means they are formed by different monosaccharide units, and most common monosaccharide units that are found in hemicelluloses are glucose, mannose and xylose. Again, these monosaccharides are linked through beta-1,4 bonds, but unlike cellulose, these components show low degree of polymerization. I will just go back to the structure of cellulose, where we discussed that it is a homopolysaccharide of glucose units that are linked through 1,4 bonds, and they have high degree of polymerization, leading to the formation of long glucan chains which are further interconnected to form fibrillar structures. In comparison to cellulose, hemicelluloses, though they have beta-1,4 bonds, but they exhibit low polymerization. Now as I discussed in the structure, these hemicelluloses, they are cross-linked with the cellulosic microfibrils, providing additional strength to the cell wall structure and preventing the collapsing or the sliding of those cellulosic microfibrils. Third component in the cell wall is of pectins. Now pectins are responsible for the rigidity of the cell wall, as well as it also protects the plant cell from various kind of biotic stresses, or it provides defense against various kinds of pathogens. It can be a bacterial pathogen, or it can be a fungal pathogen, or a viral pathogen. Now pectins are also responsible for the adhesion of two adjacent cells. Vis-a-vis, they also help in maintaining the water level of the cell walls. Now when we talk about the intercellular adhesion, these pectins act as cementing material between the two cells, thereby causing the adhesion of these to the two cells along with each other. And pectins are, again, these are the like hemicelluloses, these are the branched heteropolysaccharides, and when we talk about heteropolysaccharides, they are composed of two major type of sugars. One is the acidic sugars, which include galacto and glucouronic acid, and the other component is of the neutral sugars which are constituted by rhamnose, galactose and arabinose. Now, as we discussed that pectins are heteropolysaccharides, and as I told you that these are nothing but the cementing material that are present between the two adjacent cell walls, specifically in the middle lamellae of the cell wall, thereby helping adhesion of the two cells. Apart from the middle lamellae, pectins are also found in the primary cell wall of the plant cells, and the most common pectins that you can come across in the plant cells are homogalactouron, rhamnogalactouron 1 and rhamnogalactouron 2. So, coming on to the first type, that is homogalactouron. Now, as their name indicate, these are formed by the linear polymers of a single type of component, that is galactouronic acid units. And homogalactouron is considered as the backbone of pectins, as they constitute around 75 to 100% of the pectin structure. This is followed by rhamnogalactouron 1, which is a disaccharide, or it's a chain of disaccharide units that are mainly composed of galactouronic acid along with rhamnose. Now, these are present in a concentration of 10 to 25% of the total pectin structure. Third component is rhamnogalactouron 2, which is a very complex heteropolysaccharide present in the pectins, and it is formed by repetition of 20 different bonds of 4 oligosaccharide units, and these oligosaccharide units are formed by 12 different sugars. So, we can say that rhamnogalactouron 2 is the most complex type of pectin component amongst the 3 most common components of the pectins. And this third component of pectin, that is rhamnogalactouron 2, it constitutes hardly 0 to 10% of the total pectin structure. Now, we move on to another cell wall component, that is lignin. Now, as I told you earlier also, that lignin is a non-polysaccharide portion of the cell wall, and it has been found that it is largely made up of monomers that are derived from phenylalanine, specifically in case of vascular plants. And we know that phenylalanine is responsible for the biosynthesis of different types of phenylpropanoids, and therefore, we can say that lignin is nothing but it is a non-polysaccharide unit that is largely composed of cross-linked phenylpropanoid derivatives such as cumaryl alcohols, coniferyl alcohols or synapyl alcohols. So, as you can see in the structure of a lignin, you will see different components of cumaryl alcohol, coniferyl and synapyl alcohols that are cross-linked with each other in order to give rise to a structure of lignin. And as I told you earlier also, that lignin is largely responsible for providing the mechanical strength as well as waterproofing of the cell wall. Now, lignin is largely found, since it is responsible for the mechanical strength of the plant cell, it is largely found in the secondary cell walls, and also it is responsible for stopping the growth of the plant cell, and therefore, you can find lignin largely in certain areas of cortex. You can find lignin depositions in case of secondary thickenings of xylem vessels. You can find lignin depositions in case of fibers. So, these are few tissues where you can find lignin depositions in the plant tissues. Now, we move on to what is exactly the enzyme assisted extraction. Now, while discussing the structure of the plant cell wall, I showed you certain compounds that had interaction with certain components, whether it can be a cellulosic component, it can be a hemicellulosic component, it can be a pectin component or a lignin component. So, these compounds, certain secondary metabolites or phenolic compounds, they interact with the cell wall components and they form strong bonds with those components. We know that phenolic compounds have uh, large amount of hydroxyl groups in their structure, and these hydroxyl groups are responsible for forming strong bonds with the cell wall components. Now, when such kind of compounds are present in the plant, they are very difficult to extract with the conventional or modern extraction techniques, and these compounds remain attached with the cell wall components when the extraction is complete. So, we can say that such amount of phenolic compounds get wasted even after extraction is complete. So, to extract out such kind of compounds, enzyme assisted extraction is utilized. We also discussed that cell wall act as a major barrier in the extraction process because it hinders the entrance of the solvent into the plant cell, thereby solubilizing the components, and then it diffuses out through the cell wall. So, just imagine if we hydrolyze the cell wall, if we remove the cell wall, solvents can easily enter into the cell matrix and it can solubilize the components, and you can just imagine the increase in the extraction yields. So, enzyme assisted extraction also helps in overcoming this kind of barrier. How it does that? We will discuss it in the principle of enzyme assisted extraction. Now, since this technique utilizes different types of enzymes in order to enhance the extraction process, so these enzymes cause disruption of the plant cell wall by hydrolyzing it through or under optimum experimental conditions, and these optimal conditions will include those of temperature, pH and concentration of enzymatic solution. And after disrupting the plant cell wall or hydrolyzing the cell wall, they release the intracellular components into the solvent media, thereby increasing the overall extraction efficiency. But how they hydrolyze the cell wall? So, if I give you a brief overview of that mechanism, it includes the binding of the active side of the enzyme onto the cell wall. Now, as soon as the active site of the enzyme binds with the cell wall or forms bonds with the component of the cell wall, it causes additional stress onto the other bonds, causing the breakage of those bonds. And once such bonds are broken by the attachment of the enzymes on the cell wall, it opens certain pores in the cell wall. And once these pores are formed, gradually the entire cell wall is hydrolyzed by the action of those enzymes. So, this is how the enzymes, they act upon the cell wall, neutralizing the barrier that hinders the entrance of the solvent into the plant cell, thereby enhancing the overall extraction yield of the process. In this image, you can clearly see the effect. First, I will show you the image of the cell wall, and this is an atomic force microscopic image of a cell wall of Populus species. The first image is of topography, which shows you the surface characters, and second is the amplitude that indicates the roughness of the cell wall. Now, what happens when we incubate this material with the enzymatic mixture? So, in this you can clearly see the effect of when the powder of Populus was incubated with the endogluconase enzyme. You can just see the effect of the enzyme on the cell wall topography as well as amplitude over the period of time. You will see that there is complete disintegration of the topography, vis-a-vis there is an increase in the amplitude, and increase in the amplitude indicates increase in the roughness of the surface, which also indicates the disruption of the entire cell wall. This is another image wherein you will see just the topographic change of the cell wall of populous cells when they were incubated with the cellulose enzyme. Again, at the end of the experiment, you will see the complete disruption of the enzyme, the topography of the cell wall. This is another image where you can see the effect of enzymatic extraction on the particle or the cell wall of the fruit particle when it was incubated with the cellulase enzyme. First picture shows you the entire or the intact particle cell wall. You can see the smoothness of the surface. Second image shows you the effect of conventional solvent extraction. You can see just increase in the roughness. And third image shows you the complete disruption of the cell wall when it was incubated with the enzyme. So this is how the enzyme assisted extraction disrupts the cell wall, thereby increasing the extraction efficiency.